Okay, so today we're going to talk a little bit about artificial cells and their applications. So one of the things that I think, okay, sorry. So people were 200 nanometers, perfect. So just looking at the chat. So we're going to talk about two applications today. So one is about transport and delivery. So the best example of an artificial cell that we are kind of aware of right now and that people have been exposed is the mRNA vaccines, right? So we saw yesterday that artificial cells are basically composed of a membrane and some kind of genetic material, right? We cannot call it alive. I mean, we had the discussion yesterday. It really depends what we can call alive, but they are kind of using components from actual cells, right? So we're going to talk of one example today, which is the coronavirus vaccine based on mRNA. And so we're going to talk about this a little bit. And the second one is production of drugs. So there's a very, very nice paper that I'm going to show you today, for example, where they use bacteria to kill cancer cells. So this is actually a very interesting study that they did there, and it's just a prototype. But I feel like in the future, if we can develop this technology, it's going to be very useful for treating a bunch of diseases. So we're going to start with the just as a reminder. So we saw this yesterday. So we're going to look at it again. So this is the coronavirus. So we know that it's composed of uh, RNA genome, right? And we have these proteins outside of the virus, which are called spike proteins. They're very big, as we saw in the previous lecture, and they bind to the cell. So we saw yesterday that they bind to this ACE receptor, and once it reaches the cell, there are some proteins in the cell that basically digest this spike protein, and that creates some kind of change in the conformation. So this spike protein is very, very flexible in terms of structure. So it changes its structure very, very strongly. And in this case, it pulls up this kind of fibers that helps extend the membrane and makes possible that the virus fuses its own membrane with the membrane of the host, in this case, the human cell, right? So you can think of this kind of a pooling kind of device. So once the virus is in there, of course, it will replicate the genome and cause infection and kill the cells. So we're going to talk about the simplest model of a vaccine that uses the concepts of artificial cells. In this case, the RNA vaccines. So they're also possible to make in terms of DNA. Um, and I'm going to talk about mostly DNA and RNA vaccines. So essentially, what these vaccines are just simply we remember from the lecture yesterday that one of the things that people use for creating these artificial cells is making liposomes, right? Or kind of membranes that you can, sorry, that you can do in the lab without the need of having a cell, right? So, and this genetic material that they have for these vaccines, basically, it encodes the genes of parts of the virus, right? So we also saw from the molecular biology um, lecture yesterday that basically the RNA is the one responsible for producing the protein, right? So the RNA is the one who's going to go to the ribosome and produce the protein. So why do we need a protein? Because we need a protein that the immune system recognizes and can target the virus once it infects the person. So what are the advantages of RNA and DNA vaccines? So for now, let's focus on these two. So we saw yesterday again, it's composed of a liposome, it's composed of an RNA. So it's very fast to produce. So as I showed you yesterday, there are technologies that allows you to create nucleic acids fragments synthetically in the lab. So that makes things very easy. So what people used to do in the past is they will purify the virus and extract the proteins and use them as vaccines. But this process is very time consuming, right? And the proteins, as you saw, they are complex, they have structure. So that is very difficult. So in this case, what we are doing is we're using this mRNA that is going to go and enter the cells, our cells. The cells are going to produce that protein for us. And that protein is going to be recognized by the immune system to destroy the virus. So we're making our cells to do the job, basically. It's, as I said, it's very rapid design. So you can basically take any protein and say, okay, I'm going to have an mRNA about this virus. And this is actually very critical right now because of coronavirus. We know that a pandemic can start really fast. So it's important to have something that can be designed really quickly. The only problem is, as Miguel also showed, and we had discussion about this, mRNA is very unstable. So, you know, once it's inside of the cell, it can be destroyed very easily by proteins like RNases, so proteins that are in charge of destroying RNA, or the RNA itself. It can be hydrolyzed. It can be broken because of its structure, as we discussed before. So that means that people need several doses in order to have enough stimulation in the immune system, which is, for example, now with the Moderna and Pfizer vaccines, you need 2 doses, right? Yeah, the only thing is disadvantage, of course. I mean, for that, you need to know which part of the virus is the one that causes a strong immune response, which sometimes we don't know. So if when the older vaccines that you just give the virus, then the immune system is going to pick what is the most important part. Now we have to decide, but there's a lot of research on that. So that's also encouraging. What about DNA vaccines? Can someone tell me why not to use, well, I'm showing here the answer, no, but but if we can do RNA vaccines, we can also do DNA vaccines, right? Because the DNA is going to also give us mRNA, and it's also give us a protein. But the problem with the DNA is that it can integrate into the genome. So one important thing to clarify here is, and as we saw yesterday, the flow of information is DNA, RNA, and protein. So we cannot make, and now there's some exceptions, but in the human cell, we cannot make RNA to go back into DNA, right? Unless we have what is called an retroviral polymerase, that basically what it does is it copies the DNA into RNA. But those are things that only virus can do, right? So virus like HIV can transform RNA into DNA, but regularly our cells cannot do that, right? So it's a bit more dangerous to have something that is DNA based because it can get into our genome. But the advantage is that it's more stable than RNA. So as you know, for Pfizer and Moderna vaccines, you have to freeze. Otherwise, the RNA will be destroyed. And also the lipids will also can also degrade over time and oxidize. So it's again the same advantage as mRNA vaccines. You can rapidly design it, but as I said, but the problem is if you have a DNA vaccine and it can affect your genome, it can be problematic. For example, you know, what if it targets a gene that can cause cancer, for example, right? But that's only DNA vaccines. RNA vaccines don't have that problem, so we shouldn't be worried about it. So this is kind of an schematics of a Moderna vaccine, Moderna Pfizer vaccines. So basically what it does is we have the coronavirus and we need to figure out which protein is, you know, what is the genetic information, what are the proteins that are important for the virus, for example, for entry. And once you have that information, then you can In this case, for example, is the spike protein. That's the one that we use, that people use nowadays, people use for these mRNA vaccines. And then you can write the code and basically uh, encapsulate it in liposomes. Once you encapsulate that, then you can put it into human being, and the cells, the immune cells, in this case, the dendritic cells, are going to take that vaccine, this mRNA, produce the protein, and the immune cells are going to recognize it and are going to get activated. Once it's activated, the immune system, then it can protect against any possible, you know, infections that can happen, right? So in this case, I'm just showing simply what happens. Just a summary. Again, identify the virus, know the genetic sequence, and synthesize the mRNA. Once you have that, then you can have protection. And as we have seen, for example, if you guys have followed the news, these vaccines are very effective. So, so this is some kind of just an overview of that. So what we're gonna do today, we're gonna have a little activity. So I'm gonna introduce you to a software that is called CellPaint. So this software it's a very nice software that it can be used in the internet, or you can download the software and use it in your computer as well. So we're gonna try to use the web version. I mean, if you guys want to install it as well, please feel free. But if you have problems, then just use the web, interface. So this software is a great software for visualization of molecules. So I'm going to show you a little bit later, for example, that you can import actual structures of proteins into this software and visualize them together with membranes and the other structures, and build your own illustrations of things in a realistic way. It also has, I will show you later, kind of physics simulation involved. So meaning that you can have um, the actual simulation of the random movement of molecules in a cellular environment. So that's actually very interesting as well. So we're gonna work on this a little bit. So I'm gonna give you a short introduction to CellPaint. Um, I'm gonna try and use my CellPaint installation here, but it's gonna be exactly the same. It's gonna be exactly the same as they use in the web in the web version. And then what you guys are gonna do is you're gonna go on breakout rooms and try to do this activity. So basically, what is the activity? So we have here, I'm going to show you quickly what it is. So here we have a panel of molecules, right? So these are different proteins. For example, this is for coronavirus. So we have proteins like the spike protein in different conformations, the E protein, the nucleocapsid. We have human proteins like antibodies. We have proteins, for example, like this is the HDL, so that binds cholesterol. We have other proteins as well here, like fibrinogen, which is involved in coagulation, and insulin, for example, which is a hormone, right? We also have membranes. So we have a cellular membrane here. We have the ion channels and ribosomes that we saw yesterday. We have also enzymes, for example, that produce energy in the cell. Here we have some settings for these are the tools that we're going to use. So this tool, for example, is the paintbrush. So we're going to start simple. So we're going to draw a membrane. So basically, you just hold the click and then close it. So this creates a membrane, right? It's very simple. So here you can see, for example, if you want to add more molecules at the same time, or if you want to create more membranes. So this can help you For example, here it's not a closed membrane. Um, One thing that I mentioned you about the physics, here there's a thermometer. So you want to increase the temperature, that will increase the motions of the molecules. So you can simulate how it will behave when you increase temperature. But we're going to keep things steady right now. This tool here, it allows you to delete what you have done. If you press shift, then you can delete the whole molecule. Okay. So we have a membrane. So what can we do also with the membrane? So we can put, for example, ion channels. So ion channels go in the membrane, right? So if you put the ion channel, then it will go. Can someone tell me what's happening here? So the ion channel is pointing inwards, right? That means that when I draw the membrane, I draw it in the opposite direction. So that means that the exposed part of the membrane is inside. So in order to do it correctly, then you have to draw it in the sense of the clock. Now the receptor can go in the outside of the membrane. So if you try to draw it somewhere else, then it will say no, because it's not a membrane, right? So what else can we do? We can put a ribosome inside, for example. You see, this is a very, very gigantic protein, as we saw yesterday. If we want to know the size, we can use here the ruler. So the ruler is going to tell us the size of the, so here we have made a membrane of around 47 nanometers, which is about the size of a ribosome, right? So it makes total sense. So what about, for example, cytoskeleton proteins? So we saw yesterday that cytoskeleton proteins self-assemble. So that's actually what is happening here. So this is actin. I'm drawing basically some fibers of actin. If I increase the temperature, then the molecules are going to start to move. This tool here, it allows you to move the molecules. So if you want to kind of push something, you can just grab it and push it. So here I have pushed the membrane so strongly that it can break it sometimes. See, so you can increase here the kind of the force. So in this case, I push the membrane against the other, and you see that it's creating whole chaos. So that can be that sometimes that can happen when you push it too hard, right? And the software becomes slow. And it's because it's very hard for the physics, right? So it's becoming so random that the physics inside the software has a limit. So it becomes too slow. So then try to avoid that. So if that happens, then just simply kind of erase the problematic molecules and then just start over. So what is the activity that we're gonna do? So the activity is we're gonna draw coronavirus, and we are gonna draw the vaccine. So the coronavirus, I'm just gonna show you real quick. So remember to make the membrane in the sense of the clock. Here, okay, so it didn't, if you wanna delete something, just click here. It deletes everything. So here I'm gonna toggle physics so that it is cells assemble properly. So we're gonna put the spike proteins. The spike proteins are gonna be in the membrane, right? We can now use this to kind of arrange it a little bit better, right? We can put also some other membrane proteins of the coronavirus, and then we can randomize them. Oops. So I moved the membrane and then I put the protein away, so now it's inside of the membrane. So that was kind of a mistake there. Here we can put the nucleocapsid. So this is where the genetic material of the virus is. So I have made a mistake. I have made the membrane too small, right? So it doesn't have enough space to put the RNA. So I have to make it again, right? So we can delete this and start over. So what you guys are gonna do now is you're gonna go on breakout rooms and try to first draw a coronavirus. Now, if you wanna do the vaccine, so we have to use, in this case, a human cell membrane, as we saw before, and we have to look for the RNA. So this is the RNA that we can put inside. You can here change the color of the background. If you want to change the color of the molecules, you can also change it. So for example, here you can change the color of the RNA and then do different colors. Don't forget to toggle physics so that in this case, oh, sorry. So that it's self-assembled. Okay, so that's kind of the activity for right now. So I'm gonna close this and I'm gonna show you now the online version. So if you guys go to CellPaint, you're gonna find the link here for CellPaint, and it will load here. So this is gonna be the web version of the software. And it's exactly the same as the one that I just showed you. The only thing that it's going to be different is that you're going to be doing it on the browser rather than the software install. So here we can put also some other proteins. We can increase the temperature, reduce the temperature, and see the motions of the proteins. Okay. So once we have designed the coronavirus and the vaccine, we're gonna go here and save the picture. So this is, sorry. So here it will save what you guys, sure, go ahead. Hersh, you have a question?
Oh, yes. For when we're making the coronavirus and the vaccine, are we allowed to zoom out a little bit, or does it have to be on the same scale?
Yeah, no, definitely. Just feel free to zoom out, zoom in, do it as you like. You can change the colors. You guys feel free to do as pretty as possible.
Okay,
Right?
thanks.
Great. And then here with this picture, you can save the actual drawing, and that's what we're going to do right now. Okay. So I hope that if you guys have any questions, let me know. But I feel like it's going to be, you know, the software is very stable unless we make some problems with the physics. But as you know, if you have any problems with that, you just have to click here in the garbage bin and then start over. Okay. Or here you can delete the molecules. So if you press shift, then you can delete all the molecules at the same time. Okay. So that's what we're gonna do now. We have now, so I'm gonna stop here.